Your gas exchange and cellular respiration packets, specifically if you need help on pages 2 through 8. So these pages were the setup for an experiment that we did in class. As I go through this, you might need to pause and take time to write down things that you hear. So let's go ahead and get started. So first, this is talking about what was the same in our lab between different groups, what might be the same, and what was not the same. So first, just a little overview. In this lab, what we did is we basically took yeast, which is a living organism, single cellular, and we added sugar to our yeast, and we were interested in how much carbon dioxide gas was produced. And we were going to measure that with a balloon, and we're going to measure that by looking at the amount of foam generated in the lab. So first, let's just go through this. So what was the same? Well, very simply, the thing that was the same was the amount of yeast inside of our containers. We wanted every experiment to have the same number of yeast so we could have a better comparison. Something that might not be the same. So we certainly tried to keep water the same temperature, but that's a challenge. Water changes temperature as soon as you take it off of your heater, and we only had one heater. So something that might not be the same between each group was the temperature of the water. And then something that was definitely not the same was the sugar. That was our experimental variable, and that is what we were basically changing in our different groups. So in this box here, ways in which we could improve the design, focus on that water temperature problem, and see if you can't think of a way that we could have kept the water the same temperature in all of the groups. Page 3, this is a summary of the experiment that we did, so you can read that to get a sense. And then page 4, what you're doing here is first circling, I'd say, 2 important ideas as you read through the introduction. And then you're going to come up with your experimental question. Now, in this case, our experimental question is something along the lines of how does increasing the amount of sugar affect cellular respiration in yeast or affect carbon dioxide production in yeast? Anything along those lines would be an appropriate question. Over to page 5. Here you have a prediction sentence. So you can go ahead, you can use the sentence starter to write your prediction of what's going to happen as we add sugar to cellular respiration and carbon dioxide production. These 2 boxes you can cross out. And over to page 6. Now this box, this is where most of your work will be. And I'm actually going to start over here to the controlled variables. The controlled variables in this case were the amount of water, the temperature of the water, and also the amount of yeast. Those are things that we wanted to be as close as possible from group to group. The thing that we changed was the amount of sugar, right? So each lab group was looking at adding an increasing amount of sugar. And from that, the dependent variable, which depends on the amount of sugar, which is why it's called dependent variable, is going to be the size of the balloon and the height of the foam, which are the 2 measurements that you should have read back in the procedure. All right, as we keep going, this was the more in-depth procedure, just outlining what we did, and you should get to your tables here on page 7. Now for these tables, you'll notice table 1 is balloon size, table 2 is the height of the foam. You can find this information on Google Classroom. Under A, you'll find shared yeast data. And when you pull that up, you're going to see 2 tabs at the bottom. This is important. We have balloon data, and then we also have foam data. And so what we're going to be doing is essentially taking a look at this data. So balloon data first, and then in the blue here, that's our data for beaker 1. And then we have beaker 2, and then in the yellow, beaker 3. And then same thing over here for foam. We have beaker 1, beaker 2, and beaker 3. And then finally, looking at that data, we want to create a visualization of it. So the first one should have basically no foam and no growth of the balloon. Beaker 2 was like a medium amount of foam in a medium-sized balloon. And then Beaker 3 should have a giant balloon, the largest, and also the highest height of foam because it had the most gas production, because it was the one that ran the longest and had the most sugar in it as well. So that's going to be actually, it was just the sugar. They all ran for the same time, I'm sorry. So the one with the least sugar has the least amount of balloon change, and then the one with the most sugar has the most amount and the most amount of foam. And that's the end of this section. We'll be looking over here at sort of the conclusion on the next part, which is going to be pages, I think it's 9 through 16. But there will be a different video for that because that's part B on your lab sheet.